Hi. So for my project, I will be talking about designer babies and how we use CRISPR and Cas9 technology to create them. If you haven't heard the term designer babies yet, chances are you will soon. In its simplest definition, a designer baby is an embryo that has been genetically modified or gene edited for the sake of producing a child with specific traits. Designer babies have been the subject of much discussion and debate over the years. In particular, there have been many ethical questions surrounding genetic modifications and their purpose. Today, I'll be covering what it means to create babies in the lab, as well as the evolution of IVF technology and where it's headed in the future. So why are designer babies being engineered and how are they helpful? They're helpful as they prevent disease. In some cases, unfavorable characteristics or bad traits, like genetic disease, may be removed, or favorable traits like enhanced intelligence or strength might be added. It might help prevent genetic diseases such as Alzheimer's, Huntington's disease, Down syndrome, and spinal muscular atrophy. The modification can also reduce the risk of inherited medical conditions such as anemia, obesity, diabetes, cancer, and much more. Individual traits can be selected to the preference of the parents. This leads into ethical questions, which we will discuss on later. So how is the genetic modification carried out? The idea of genetic engineering in terms of designer babies all stems from in vitro fertilization or IVF technology. In many ways, designer babies are the obvious result of decades of advancing IVF technology. Once scientists discovered how to create babies in the lab, embryo editing to produce a healthy GMO baby was perhaps a natural next step. Scientists saw the potential to not just optimize genes for disease prevention, but also choose aesthetics and personality traits. There are various technologies involved in the creation of a GM baby. One protocol is pre-implantation genetic diagnosis, where the embryonic genetic defects are identified pre-implantation, and only embryos devoid of certain genetic disorders are implanted. However, the technology I will be focusing on today is CRISPR genetic engineering, which was originally created in the 1980s. Cas9 is a naturally occurring protein which works with CRISPR technology to modify the genes. The significance of utilizing this technology to our advantage in the lab with genetic engineering, however, was only relatively recently understood. CRISPR babies are created by modifying DNA fragments to prevent and correct disease-causing genetic errors. So the CRISPR-Cas9 technology was first identified in E. coli bacteria. It stands for clustered regularly interspaced short palindromic repeats. The first part of that name, clustered regularly interspaced, means that there are unique spacer DNA in between the palindromic repeat sequences. This spacer DNA matches with the multitude of viral/bacteriophage DNA, which thereby means that spacers are basically the history of old infections that the cell won't be infected by again. The second half of the name, short palindromic repeats, basically says that there are palindromic repeats in their DNA that is a sequence of DNA that is read the same right and left. So this is the CRISPR system in bacteria cells. To the left, you can see the Cas genes, which produce helicases and nucleases. And to the right, you see the CRISPR system. So in a situation where the virus is coming in and injects its viral DNA into the cell, the DNA will produce a Cas system where which holds the corresponding spacer DNA, and when that comes in contact with the viral DNA, the whole complex breaks down the viral DNA and creates a immune system for the organism. So Jennifer Doudna and M.A.U. Charpentier were the first ones who brought CRISPR into humans. They used S. pyogenase and the Cas9 protein from that bacteria. The Cas9 protein is a nuclease that can cut DNA. And so this is basically the CRISPR-Cas9 system that is used in humans. It has a matching CRISPR RNA that matches the sequence of DNA that we want to cut, and then we have the tracer RNA that holds it in place, and the whole structure is called the guide RNA, and we also have the protein Cas9 that does the cutting. So to break a gene in humans, first the genetic engineer identifies the specific DNA sequence that we want to inactivate. Next, the guide RNA strand that matches the sequence will attach to Cas9. The DNA will feed into the protein, and then the enzyme will cut. After the cut is made, the cell will try to repair the break, and thereby the gene will be inactivated. To insert a gene, there is a three-part system: the Cas9 protein, the guide RNA, and the host sequence that we want to put in. It's the same process as breaking the DNA, but instead of letting the cell repair the break, we insert the DNA that we want. Now, onto the negative aspects of this type of genetic modification. The technology used is not 100% safe yet. It is only in the experimental stages at this point. Because the technology is so new, it is unknown whether the babies will affect the gene pool. This can cause difficulties later on throughout the baby's family tree. Next, a baby cannot consent to have its own body altered. Therefore, some do not believe it's right, as parents do not own their children, argument that is unethical. Additionally, parents may use this technology for superficial purposes, such as purposely seeking out a blonde-haired, blue-eyed baby for appearance concerns only. This leads into the concern that because most people will seek out good-looking, intelligent babies with other optimum characteristics, everyone will be relatively similar, therefore there would be no individuality. Finally, this procedure is not cheap. In vitro fertilization itself costs $15,000, so the amount to modify the embryos is expected to be quite high as well, and not everyone would be able to afford it. So that was my medical technology presentation, and I hope you enjoyed it.